U gaat staan. U kunt op plaatsen.
Hereby I open this academic ceremony in which Anaís Sánchez Castillo will defend the academic thesis Sertraline-mediated targeting of serine and glycine synthesis in multimodal cancer treatment, the impact of a repurposed drug on cancer cell metabolic adaptation and the tumor microenvironment in het openbaar zal verdedigen. Very long title. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you very much. So, dear Prorector, dear members of the Corona, dear supervisors, dear family, friends and colleagues, thank you for being here today. In the upcoming 15 minutes, I will present you with a summary of my thesis and the research I conducted during my PhD. First, I would like to introduce you the concept of cancer. To understand cancer, we need to start by recognizing that the human body is made up of different types of cells. These cells are organized within our body to form the different organs. If we take a closer look within these cells, we discover that inside each cell there is a book of instructions called DNA. This DNA contains genes that are like the individual recipes in the book, and these genes are written with letters called nucleotides. These genetic instructions are used to make the proteins that are like the workers in a factory, and the factory is the cell. And these proteins ensure the proper functioning of the cells and the organs within our body. However, in cancer, these instructions undergo an accumulation of mistakes. And when this happens, the protein workers start behaving abnormally, causing uncontrolled cell division and growth and the development of cancer. In addition to the uncontrolled cell division and growth, cancer cells have unique characteristics that make them different from normal cells in our body. These cancer cells have the ability to evade, adapt to, and also survive the body's defenses and all the cancer treatments that we use to eliminate them, which causes the failure of cancer treatments. Therefore, it is important that we identify and block these survival tricks and mechanisms of cancer cells in order to enhance the effectiveness of cancer treatments. One of these specific characteristics of cancer cells is their altered metabolism. Cancer cells are very hungry, they crave more sugar, being on a constant sugar rush. This is driven by changes in these genetic instructions and the protein workers, which start increasing the consumption of nutrients and also the metabolic activity of cancer cells. All these in order to meet the high requirements of nutrients that cancer cells have. For this, these protein workers start being organized in various metabolic pathways or so-called departments. And these departments are responsible for the production of the different building blocks that cancer cells need for their growth. These building blocks are, for instance, proteins, nucleotides, and antioxidants. But these building blocks are not only important for their growth, for the growth of cancer cells, but also for their ability to survive and resist treatments like radiotherapy. Radiotherapy causes the damage of the DNA, these cell instructions, and create stress within the cells with harmful molecules called ROS. These ROS molecules further induce damage within the cells. But the cancer cells that survive do so because they can increase the production of nucleotides, the building blocks needed to repair their damaged DNA. And also because they have enough amount of antioxidants, which act like vacuum cleaners within the cell, cleaning up these harmful ROS molecules. During my PhD thesis, I focused on investigating one specific metabolic pathway or department, that is the serine and glycine synthesis. This department uses the sugar consumed by cancer cells in a chain reaction carried out by different workers in order to produce finally serine and also glycine. And why were we interested in investigating this metabolic pathway, and why are serine and glycine important? This is because serine and glycine are necessary to produce the building block, many of these building blocks that cancer cells need, and we previously mentioned, such as nucleotides, but also antioxidants or proteins. We investigated this metabolic process in lung cancer. This graph here shows the levels of the different workers involved in the synthesis of serine and glycine, comparing normal lung cells and lung cancer cells from lung cancer patients. We found that while the levels of these workers were low in normal cells, lung cancer cells had higher levels of these workers. This indicates that the high synthesis of serine and glycine is a characteristic of cancer cells and not the normal cells. In addition, we also explored how these lung cancer cells respond to radiation therapy in the laboratory. We specifically looked at SHMT, this protein worker involved in the last step in the production of serine and glycine. We irradiated lung cancer cells in the lab, and what we found is that these lung cancer cells, with increasing doses of irradiation, they also increase the levels of SHMT. In mice models, tumors that were irradiated, here shown in red, also showed higher levels of SHMT compared to non-irradiated tumors in gray. This suggested to us that lung cancer cells increase the levels of SHMT and also the production of serine and glycine in order to recover and survive from the radiation therapy. But this is not unique to lung cancer. We also investigated this metabolic process in glioblastoma, which is a very aggressive type of brain tumor. We looked specifically at PSPH, this protein worker here. Looking at PSPH in glioblastoma patients, cancer cells had higher levels of this PSPH in red compared to normal cells in blue. When we analyzed the genetic changes, the changes in these DNA instructions for this protein, we found that approximately 12% of patients present PSPH amplifications or extra copies of this gene, and approximately 1% of patients present a PSPH mutation or mistake. To investigate what are the consequences of these alterations in the PSPH protein, in the lab we created glioblastoma cells that were mimicking the patient's cases with more PSPH copies and the PSPH mistakes, here shown in red as the high PSPH and high PSPH mutated. Then we measured the production of different nucleotides that are the building blocks of the DNA, the cell instructions, and we found that compared to control cells, those cells with high PSPH and high PSPH mutated were able to produce more nucleotides. Their production was higher, shown here in red. Therefore, recognizing the importance of serine and glycine for the production of building blocks for cancer cells, for their growth, but also in response to treatments like radiotherapy, our aim was to block the production of serine and glycine. To achieve this, we used a compound called sertraline. But sertraline is a repurposed compound. It's actually used already in the clinic, but as an antidepressant. However, our group has demonstrated that sertraline is also able to block the production of serine and glycine. It could do so by blocking this step here of the process. We investigated then in the lab the effects of sertraline treatment in lung cancer cells that were exposed to different doses of irradiation. This graph here shows the survival of these lung cancer cells in two different conditions. In blue, the cells that were only irradiated with increasing doses of irradiation, and in green, the cells that were treated with sertraline in combination with increasing doses of irradiation. And we observed that those lung cancer cells that were treated also with sertraline showed a lower survival, a reduced survival, compared to cells that were only irradiated. In mice models, tumors were treated with radiotherapy, sertraline, and the combination of both, here shown in green. Those tumors that were treated with a combination of both, radiotherapy and sertraline, showed the lowest tumor weight. Those tumors were smaller. These results indicate that indeed lung cancer cells require serine and glycine in response to radiotherapy, but blocking their production with sertraline could reduce the survival and also the growth of those cancer cells. Then, the identification of this high serine and glycine synthesis as a characteristic only of cancer cells and not the normal cells necessary for their growth and survival in response to radiotherapy provides the opportunity to block this process with sertraline in those cells, selectively eliminating the cancer cells, while at the same time it could also reduce the damage to normal cells and therefore the side effects of cancer treatment. But cancer cells are not alone. They are part of what is called the tumor microenvironment. It's like the neighborhood where the tumor exists. And the metabolism of cancer cells is also important in how they interact with these surroundings. For instance, cancer cells that produce more serine and glycine can also release this serine and glycine to their environment and influence the behavior of other cells there, such as immune cells. And what are the immune cells? The immune cells are the security guards against cancer. They are within our body, able to recognize and eliminate cancer cells. However, cancer cells again now develop strategies to make it more difficult for the immune cells to detect and destroy them. One of these strategies that cancer cells use is to send signals, and these signals are able to confuse and block the functions of the immune cells, which will help them escape and survive. One of these signals is galectin-1. During my PhD, we found a connection between serine and glycine synthesis and this blocking signal, galectin-1. In glioblastoma cells with high PSPH, with more PSPH copies and PSPH mistakes. We found that compared to control cells, cells with high PSPH and high PSPH mutated had higher levels of this blocking signal, galectin-1. This indicates that these cells can use galectin-1 to block the immune security guards, and they present then another survival advantage. However, when we blocked the production of serine and glycine using sertraline in combination with radiotherapy, we observed that the levels of this blocking signal, galectin-1, were reduced. This suggested to us that with this combination treatment, the immune cells were able now to recognize and eliminate the cancer cells. To conclude, during this presentation, I've shown that in different types of cancer, cancer cells show high synthesis, high production of serine and glycine. And this is one of the specific characteristics of cancer cells that they use to grow and also to survive treatments like radiotherapy. But blocking the production of serine and glycine with sertraline in combination with radiotherapy could reduce the levels of these building blocks that cancer cells need for their growth and survival, and also could reduce the blockage of the immune cells that could now recognize and eliminate the cancer cells. All these then leading to reduced survival and growth of cancer cells. As future perspectives, it will be important to identify those patients with tumors with high production of serine and glycine, because those will be the ones that will most likely benefit from sertraline treatment, blocking this high serine and glycine synthesis. But this is particularly interesting in combination treatments, for instance radiotherapy in lung cancer, to overcome the resistance and survival of these cancer cells to the treatment, or with other repurposed drugs such as chloroquine that has been also discussed in chapter 5 of my thesis and represents another therapeutic strategy that we are investigating. With that, I would like to thank you for your attention and give back the word to the prorector.
Thank you very much for a very clear presentation. It's now time to start with the opposition, and the opposition will be opened by the chair of the assessment committee, and that is Professor Here, Professor of Molecular Imaging at Maastricht University.
Thank you, Prorector. Dear candidate, before I start asking my questions, I want to give you a huge compliment, not only with this wonderful thesis that was a pleasure to read, but also by stressing the importance of metabolism. I think sometimes we underestimate that, and particularly linked to the multimodal treatment, I think that's one of the right ways to go forward. So, of course, with those compliments, and not only to you, but also to your supervisory team who has stood by you along the way. I want to start you off with a somewhat more generic question, a little bit along the lines of your presentation. For your studies, you've used patient-derived cell cultures. You already mentioned that there is an environment and other cells, immune cells, but yet you study these cells in isolation. Is that the right strategy? Should you not study these cells in their native environment and look for that metabolism when these cells are actually talking to their environment with the other cells, with the signals that are sent out? Would that not give you a much better insight in what is going on? And if your answer is going to be yes, you're absolutely right, then how would you do that?
Highly esteemed opponent, thank you for your kind words and your question. So indeed, throughout my whole thesis and mostly in the discussion, I mentioned that indeed it will be important to investigate tumor metabolism, but also the interaction with the immune cells and the other cells in the tumor microenvironment. And indeed, I didn't do that. But what I think it's always not the way to do that would be with mouse models, with immunocompetent mouse models, for example, that have the immune system. But it's also not ideal because it's also mouse metabolism. So that's why what I propose is to first of all, try to use culture conditions that more replicate the concentrations of nutrient and oxygen, for example, that is in the tumor microenvironment. Because most of the times the cell culture media that we use have higher or lower concentrations of the metabolites and nutrients that are in the environment. And also maybe co-culture experiments and assays could be another way to go, at least to have a better idea what the interaction between them is, and mouse models, but also not ideal because, as I said...
every method, I agree, every method has its challenges and drawbacks. I agree, I probably would also go for some co-culture model. But there's one other aspect that in your analysis that might give a slightly skewed picture, and that is you homogenize all of your cells. And when you culture your cells, they're all perfectly synchronized, they're in sync. But in a tumor, they're all in different states, and you have immune cells in different states. Could you think of a way where you could account for that heterogeneity in your studies? Maybe with mouse models and maybe even in immune-compromised mouse models, because obviously if they're immune-competent, it's going to be a little bit more difficult to grow the tumors. But curious to learn what your thoughts are on that.
So how to overcome how to also mimic the heterogeneity that happens in the lab. So I could think maybe using patient-derived material, so you get a tumor biopsy with heterogeneous population of cancer cells, and growing them in 3D or in organoids, and then maybe use the co-culture with the immune cells.
So how would you then study the metabolism? Because then you want the metabolism for every single cell, right? So how would you do that?
Maybe special metabolomics could be a way to go. For example, single cell sequencing also, and more moving towards the single cell high.
I think indeed spatial
methods.
Biology and spatial metabolomics, I think that's one of the elements that I think you could really benefit from, that understanding of the cell-cell communication and the effect of your, indeed, the tumor microenvironment and the effect of your different combined treatments. But in that, so let's assume you do that. I think that's a great idea. We could help. But then there's one other element that I'm missing in your thesis, and the other elements, and you mentioned that your soldiers or your workers, the proteins. I read all of these beautiful results on the metabolism, But then I was looking for, okay, what are the protein pathways that are involved? Because they're intricately combined. It's not these 3 proteins, but they're part of a network, as you're well aware of. Could you elaborate a little bit more on, why is the proteomics missing, but also how would you approach this if you would have access and more time, because time obviously is of the essence. How would you incorporate that?
If I understood properly, is to identify also the protein drivers that are driving, like the metabolism.
So you talk about metabolism and you draw all kinds of conclusions, but you say nothing about, okay, what's actually happening. So how certain are you that your conclusions are well substantiated because you have not looked yet at the protein? So what is missing? How would you approach it? How you would solve that problem?
So indeed, the thing with this metabolic pathway is that there are a lot of genetic drivers and signaling pathways that are regulating also this. In different cancer types is also a different mechanism, but also there the heterogeneity also again plays a role, of course. Maybe continuing with the spatial metabolomics, this could also be combined with some proteomics too.
So spatial proteomics in that would be ideal, but even
also
already
proteomics by themselves, yes.
Exactly.
Themselves.
On your extract, yes. As you've done your metabolomics, it would be nice to coordinate those networks. I'm looking at the prorector. Have I got time for one short question? Yeah. Then my last question before I give the word back to the prorector is actually on the impact of your work. And you write this beautiful chapter on the impact of your work where you address scientific impact and societal impact. And while I understand your societal impact, I'm missing the economic impact. Particularly if you think about these new treatments and these combination treatments with radiotherapy, I mean, there's a lot to be gained for healthcare. Could you elaborate a little bit on your vision on the economic impact of your work? And particularly, I want to make a distinction between the short-term economic impact, if it's there, and the long-term economic impact.
Yes. So in short-term economical impact, first of all, we are using a repurposed drug, so then it's is cheaper than newly developed drugs, and it's already FDA approved. And also by combining treatments, we could reduce the dosage potentially of chemotherapy and also radiotherapy, because it's like a multimodal. The idea is to do a multimodal cancer treatment. So this would be the short-term economical aspect. And then in long term...
And then the short term, because that's cheaper, you use less drugs, right?
Yes, exactly. And then in long term, if we can improve the outcome of patients, better understand that, and also with less side effects because of using like these combination treatments, and also with these metabolic vulnerabilities of cancer cells that we might target more the cancer cells, then we also could reduce the side effects and then the long-term effects of the side effects of the treatment, and then for more long-term economical impact.
Okay. So I assume that you mean that economical impact is because you actually essentially extend the lifespan of patients much shorter, and in our growing aging population, we need our workforce, so you actually contribute to the workforce of the future. So I'm very glad to hear that. And with that, I'll give back the word to the prorector.
Very optimistic thought. The opposition will be continued by Dr. Korycinski, who's present online, I hope.
Yes, thank you.
There you are. Dr. Korycinski is assistant professor at Princess Margaret Cancer Centre at the University of Toronto in Canada. Floor is yours.
Well, thank you. I'd like to start by congratulating the candidate on a very comprehensive, interesting, and well-presented thesis. I'd like to start by discussing with you, dear candidate, some of your work in the context of what else has been done in the field and what we think as the conventional wisdom in some of these signaling pathways. So I think the conventional wisdom is that HIF, important transcription factor in cancer and in hypoxia, upregulates enzymes in the serine synthesis pathway, and stimulates serine glycine synthesis. Do you feel that your results are someone in contrast to this conventional wisdom? And if so, what would you hypothesize is the underlying mechanism of why some of your results may be different than what is found in literature?
Opponent, thank you for your kind words and your question. Indeed, in chapter, I think it's 4 of my thesis, we investigated the knockouts of HIV genes, HIV1 and HIV2, in normoxia, not in hypoxia. And we first expected, indeed, to have less synthesis of serine and glycine, because HIV has been described to increase the synthesis in hypoxia. So we were expecting to actually see the opposite. So that was surprising. But it's true that we are looking at this in normoxia. So might be that is a different mechanism in normoxia than hypoxia, that somehow in normoxia this is actually repressed, mediated directly or indirectly by HIV, and then in hypoxia it gets upregulated. But it's true that we don't know yet what is the mechanism behind. So now at this moment I cannot answer what is exactly the mechanism, but I've been checking also. We know that HIV binds to the promoter region, for example, of SHMT, one of the enzymes of the pathway. And also I've been reading, for example, that HIV3 is not well investigated, but can act as a repressor, one of their isoforms. So could be that somehow they are linked. Also, this one is upregulated in normoxia and downregulated in hypoxia, so could be, but I cannot know for sure. So maybe some proteomics, so the idea would be to do some proteomics in those cells to see what is upregulated or downregulated, to see if we can find a link between the two. But,
thank
opposite,
you. So,
yes.
Yes, thank you. And so, so if you have the time and the resources, what would you do to corroborate this theory that perhaps the discrepancy is due to experiments in normoxia versus hypoxia, and to convince the world that HIF restrains but not stimulates serine glycine synthesis?
Okay, so if I could continue with the project, the first thing I would like to do is indeed proteomics and maybe some more metabolomics to have a better idea what is happening in these cells. So then we can proceed to better investigate that in the two conditions, normoxia and hypoxia. Maybe using also again some knockouts depending on the results, and also maybe some in vivo assays where we can have also more heterogeneous tumor with normoxia and hypoxia, and better investigate the differences between the two. Yes, I would think so. Yes.
Okay. Well, I think proteomics and metabolomics are good discovery tools. I think there's probably experiments that could be designed in a more hypothesis-driven way as well. But would be interesting to do. I had a question about, this conventional wisdom. Let's say that HIF stimulates serine glycine synthesis. We think of an important job of HIF is to shut down TCA and electron transport chain activity in hypoxia through upregulation of PDK1. So, do you find it paradoxical that HIF would stimulate serine glycine synthesis in that context, since that pathway actually fuels TCA activity and electron transport chain activity by producing NADH and alpha-ketoglutarate? Do you find that to be a paradox?
Okay, let me think for a bit. So, if the activation of serine and glycine synthesis by HIF in hypoxia is a paradox, because during hypoxia then it's cells go to oxidative phosphorylation but serine and glycine synthesis also has been shown to be related to the oxidative phosphorylation when knock down, for example, SHMT, that is the one I investigated. Also, deficiencies in oxidative phosphorylation also happen. Wait, no, but this is exactly it. Yes, so could you rephrase the question in a different way?
Yes, I'm wondering if it's a paradox that HIF at the same time will shut down conversion of pyruvate to acetyl-CoA to prevent TCA activity, but at the same time stimulate alpha-ketoglutarate production to fuel the TCA cycle.
But this is, with the knockdown of HIF, we saw the increase in alpha-ketoglutarate.
Right, I'm talking about the results in hypoxia of other investigators.
Yes. Yes, but yes, so this indeed could be something to investigate further. It's indeed a paradox, but now I don't know how the cells could benefit from that at the same time. Sorry. No.
Okay, that's fine. Let me ask you one more thing about HIF. So you found that HIF1 and 2 seem to have quite different effects in serine glycine synthesis and also in the response to radiation. Can you just speculate for us what the evolutionary purpose might be for HIF1 and HIF2 having such different roles?
Thank you for your question. So indeed, when we knock out HIF1 and HIF2 under normoxia, we see different phenotypes and metabolic requirements for those cells, so indicating that they have different purposes. And evolutionary, I can think maybe of like they have some differences in the tissue they are expressed, so maybe depending on that, depending on the expression tissue, then they can might require some or others metabolites, and that's why they have different functions.
Okay, thank you. Pro-Rector, do I have time for one more question?
I'm afraid your time is up. So we have to continue. Thank you very much for your questions. The next opponent is Professor Sanvaux, Research Director of AREC and Extraordinary Professor at the Université Catholique de Louvain in Brussels.
Thank you, Pro-Rector. Dear candidate, I extend my congratulations for your work. I enjoyed reading it. I would like to go in metabolomics directly and to ask a question about how can you differentiate the production versus the utilization of metabolites by combining the C13 labeled experiments to the absolute level of metabolites.
Highly esteemed opponent, thank you for your kind words and your question. So your question is how we can differentiate production from consumption? Okay. So for production, we use, in my thesis, we use the glucose tracing. So we give leveled glucose to the cells, and then we follow the carbons to see if this glucose is used for the synthesis, so we can see if they produce different metabolites. And for the consumption, we have the media, we measure the metabolites in the media, and then we can see if they are reduced in this media, and also if we can follow the labeled metabolites to the media, so we can also see if they exported to the media as well, because they are labeled from the synthesis. Yes.
Thank you. Could you differentiate a metabolite, an intermediate metabolite that is produced very fast but also consumed very fast, from a metabolite that is not produced? How can you, or little produced?
Oh yeah, for this we need the fluxes and time point to do that. So in my case, we could not differentiate the flux, only the levels, intracellular level synthesis, but not really like fluxes and their timing of production and.
According to you, the expression and the activity of given enzymes in the serine pathway, for example, is it important in addition to the metabolic measurements that you made? Because I saw very few Western blottings and very few activity tests on specific enzymes.
Yes, thank you for your question. So indeed, for these studies, it's important to not only see if the expression of the enzymes of proteins is changing, but also their activity. So for that, indeed, the glucose tracing to see if there is, for example, more synthesis, that would indicate that there is more activity because it's being more produced. But I also believe that if there are changes in the expression gene and protein, it might indicate that those cells are increasing the expression also for a reason. But indeed, the combination of both is important, also the activity, not only the expression.
And during your thesis, you measured serine and glycine metabolites in the plasma of patients that had been irradiated, right? And you said that there were lower levels of these metabolites in the plasma because it showed that these metabolites were used, right? And so here again we are in a question about production versus use, because when you irradiate cancers in patients, you kill cells, so you kill the producers. How can you be sure that the decreased level is due to the use of these metabolites versus a lower production of these metabolites?
Yeah, we can, indeed, can be both. Can be that they produce less and therefore less export to the plasma, to the environment, and also that they consume more, so it can be both. So there we cannot distinguish that. That's why we continued, try to investigate in vitro if it was that cells in response to radiation try to produce more or consume more. But this is what suggested to us that they were consuming more, but can be both indeed, okay. Production and consumption.
Then if we move to the financial aspect of your thesis and the economical aspect, Sertraline appears to be at the core of your work, an FDA-approved drug that could be repurposed. How sure are you that Sertraline is really sensitizing tumors because of SHMT inhibition? How selective is that drug?
Thank you for your question. So sertraline has been investigated also in other studies to have anti-tumor effects, not only because of SHMT, but in those studies they never identified like a target like sertraline to bind, for example, to the catalytic bind pocket of the enzymes, but more they saw differences in signaling pathways like P53 or mTOR. And autophagy, but never to. They didn't identify like binding and inhibiting another protein, but of course could be other off targets. But what we see along our studies is that those cells that have higher synthesis of serine and glycine, or for example with the PSPH overexpression, they are also more sensitive in most of our studies to sertraline. And also, not only in that study in my case, but also in others that our group has investigated before.
If we admit that SHMT is the main target of sertraline for radiosensitization, how would resistance occur?
Resistance?
Is it possible that resistance would occur, and how?
I can imagine that cancer cells would try to increase synthesis of nucleotides and antioxidants by other pathways and mechanisms. So
could they also act on the expression of the enzymes? Could the cells change the expression of SHMT1 or 2 or both?
Within the same pathway? We haven't seen that, but of course could be something that we could investigate. But we, even if they increase the expression of this pathway, then we are also inhibiting with sertraline. But Also in our study, we saw that long-term, for example, in chapter 2 in non-small cell lung cancer, we had a long-term experiment to see if the cells could also regrow and acquire resistance to the treatment after we removed the treatment for already, I think, 20 days we had it growing in spheres, and they didn't start growing more or develop resistance. But of course, this is something that needs to be investigated, of course, if there are resistance mechanisms.
And to connect
treatment.
the first and the second part, I mean, the chapter 3 and 4, hypoxia could play a role in resistance to sertraline. Is it possible?
Yes. In hypoxia, the serine and glycine synthesis is upregulated. So I imagine that also tumors that have been treated with sertraline in those hypoxic areas, they will also increase the expression and the activity of serine and glycine. So we could also target it with sertraline. But long-term needs to be investigated, of course, if after the treatment, long-term, we see some resistance mechanisms.
Thank you very much.
The opposition will be continued by Professor Lambin, Professor of Precision Medicine at Maastricht University, and he's present online. Professor Lambin?
Yes. So first, I would like to express my congratulations for your work, which has several facets: radiobiology, metabolic studies, and even drug development. And I also would like to take this opportunity to congratulate the supervisory team, and I could recognize the signatures of the three of you in this work. My first question would be about the dosage, the dose of sertraline that you have used, because we have seen with your collaborator, your supervisors, Dr. Rushop, that with chloroquine, that having the right dose is actually not always easy in drug repurposing. So my first question would be, which dose have you used for a concentration of sertraline have you used for your in vitro and in vivo work, and why?
Opponent, thank you for your kind words and your question. So in our studies with sertraline, because of the combination treatments, we always try to use like lower concentrations, so that as monotherapies would not have any effect. And in vivo studies, we always used an equivalent concentration.
In micromolar, how much is that?
Yes, so around between 5 micromolar, 5 and 10 micromolar, maximum 10, but between 5 and 10 micromolar. And in vivo studies, we used an equivalent concentration to what the patients as an antidepressant take as maximum dose. So the maximum dose is 250 milligram that patients take as an antidepressant. So for a person of approximately 70 kilos, is like 11 milligram per kilo, but they take it daily. And in our in vivo studies, we used 15 milligram per kilo, and we used every 3 days, in a space of 3 days. So equivalent doses.
Right, you said that the maximum dose was in the clinic was 250 milligram. And what is the corresponding Cmax of maximum concentration obtained in the plasma with that dose? Do you know that?
Now, I don't remember on top of my head. I know that the concentration maximum they acquired after 6 hours after the administration, but I don't remember now exactly what was the concentration. Sorry.
Yeah, because I just found in the literature that would be around 0.6 micromolar, so quite significantly lower than the 5 to 10 micromolar. So does it mean that if you want to use that in the clinic, you will have to do some dose escalations or do sort of a phase 1 trial? How do you see that?
So even if it's a FDA approved drug, we need to still have the phase 1 with, because we are also going to combine it with other treatments, so to see their toxicity and also to see the concentration that we see that is more effective. So yes, I still think we still need to go through that to see.
Yeah, and do you have any idea of the concentration you obtain in your animals with the dose administered? Did you do any pharmacokinetic study? Or?
So these now I also do not remember on top of my head what was the concentration.
All right, so let's go to our next question. In our department, we work on different disease, and we know homocysteine because it's a biomarker for cardiovascular disease and also even for dementia. So I noticed that the word homocysteine was cited 25 times in your thesis. So could you elaborate? What is the meaning of a high or low level of homocysteine in patient for the serine glycine pathway? Is it a potential biomarker, and is there any potential interest to try to modify the level of homocysteine?
So homocysteine is downstream of serine and glycine synthesis. So serine and glycine conversion donates 1 carbon units to this 1 carbon metabolism, and homocysteine is part of that. So I hypothesized that indeed, if there is more serine and glycine synthesis in those cells, that also this homocysteine might be altered. And also when we block, actually, okay, let me see. When we block in chapter 3, with sertraline in combination with radiotherapy, what we see is that glycine levels in the plasma goes down, but the homocysteine goes up. It's like we block also this metabolic process there, so homocysteine cannot continue, like how to say, their process. So could be indeed a way.
Would it mean that a high level of homocysteine would be a positive biomarker, or is the reverse?
This would be this still we need to investigate it, but according to this study in which we blocked the production with sertraline in combination with radiotherapy, we saw increased levels of homocysteine. Like there was an accumulation, and that could be maybe used to see more like a response thing, more than a biomarker in this case, to see if the treatment is, if the cancer cells are responding to the treatment. And also the other case that we saw was with radiotherapy. Indeed, I think we saw decreased levels of homocysteine. So could also be more like a response of the treatment than a biomarker. This will need to be investigated, but of course, but they are related to each other, so that could be a good. And then we detected in response to treatment, so that could be a good point to investigate to see if we can use it as biomarker.
Excellent.
And maybe
Thank you for your answer. And I would like to ask your paranymphine to read statement 3, please. Excuse me, statement 5. Statement 5.
5? Okay.
5, yes?
Investigating the metabolic crosstalk between cancer and immune cells is critical for enhancing the efficacy of metabolism target therapies while simultaneously inducing an anti-tumor response.
Could you keep your, Professor Lambert, could you keep your question short and a short answer, please?
Yeah. Could you elaborate and tell us whether sertraline would also inhibit the effect of immune cells?
If sertraline could inhibit the function of immune cells? Yes. So there are different things to consider. So first of all, serine and glycine metabolism in immune cells. So it has been demonstrated that serine and glycine synthesis or uptake is also important for immune cells. So then if we target that in the tumor cells, we might also be targeting that in the immune cells. So it's something that needs to be investigated. For example, has been shown that serine uptake is important for the proliferation and expansion of T cells, but also related to some immunosuppressive macrophages phenotypes. So more serine and glycine synthesis, more immunosuppressive. So that part needs to be.
Okay, so last question with just an answer, yes or no. Because non-small cell lung cancer receive also immunotherapy, would you then give sertraline during concomitantly with immunotherapy non-small cell lung cancer, yes or no?
Yes, I would.
You would.
Sorry?
Would you then give sertraline concomitantly to immunotherapy in non-small cell lung cancer?
I would need to
Very short answer.
investigate it.
Okay, thank you very much, Professor Lambert. Sorry to interrupt the discussion, but there's 2 more opponents to go. So the next opponent is Dr. Nabbe, Assistant Professor at the Department of Genetics at Maastricht University.
Thank you. So yeah, first of all, I would also like to congratulate you and your team on this really impressive thesis. And I think your family, who came all the way from Spain, they can be very proud at all the work that you accomplished over the last couple of years. But yeah, of course, I read it with pleasure, but I also have some questions, of course. So my first question is, actually, do you enjoy soccer?
Soccer?
Yes, football. Do you enjoy that?
Yes.
Yeah. And are you familiar with the game itself? Do you know how it works?
Not the rules, just the fun of it, but not too much.
But the basic principles you know what
yes,
it's like. So if you would start a soccer game and the ball is not thrown into the field, what would happen when you think about the players?
So, if the ball doesn't touch the field.
Yeah, if it's not getting into the field, there's no ball in the field.
We need to move the ball to the field to start playing.
Yeah, because otherwise not much is happening, right?
Yes.
So when you now go to your pathways that you're thinking about, what is on the top or at the beginning of the pathways? Of your glycolysis pathway? What is at the start of glycolysis?
Glucose.
Yeah, and if we now go to your chapter 4 on page 127, and then we go to figure 1b.
1b.
Yes.
Yes.
Where you have the intracellular levels of glycolytic metabolites. And I look at your HIF2 alpha knockout line. Is that glucose? Or it seems there's no, right? Or it's very low, or there's...
Very low, yes. The intracellular levels. But that could mean that that is being metabolized, that the intracellular levels not metabolized are low. But
okay, so
it's
because
not
you didn't say anything about it, right? But you say that, for example, you have 2 alpha knockout line, they show a 2-fold reduction in fructose, 1,6-biphosphate. They see an increase in synthesis of the serine. But so it means all the glucose is gone then? It's all going to that pathway and there's no glucose left? Because in your HIF1 alpha knockout, you do see glucose levels, right? They're actually higher than
yes.
the control lines. Do you have any explanation for that?
I could think that in HIF2 alpha knockout, the glucose is being metabolized for downstream pathways, and that's why the levels, because this is intracellular levels, are lower, and shows again the difference between HIF1 alpha and HIF2 alpha knockout in line of regulating this glycolysis or metabolic pathway.
Okay, so it's all being used, basically.
I assume yes, but it's the only thing I can think of now.
Okay. And, as you know, I mainly study substrate metabolism in the heart, right? The glucose and the fatty acid substrate metabolism. We're also looking at metabolic switches, and usually what we do is we try to intervene at the level of the substrate transporters. So could that possibly be a target for you? Like if you could you prevent the glucose from coming in, for example, into the cell, or if you go more upstream in your pathways, could that be something to intervene?
So the advantage of targeting the synthesis in this case was that it is upregulated in cancer cells, so we could target this vulnerability more in cancer cells. About transporters for the import of, because serine and glycine can also be uptaken from the environment, about their uptake, I didn't see papers so far, publications that show an upregulation of the transporters, but most of the times it's increase of the synthesis, because with the uptake, cancer cells do not have enough, and then they also increase the synthesis. And by blocking the transport of glucose, I think what I see, the problem might be also the toxicity for the normal cells as well, not only for the cancer cells.
Okay, and I'm also looking at the time in the meantime. I will make a last question then. I was just wondering if, would you consider the alterations in metabolism as a cause or a consequence for cancer? Alterations as a cause or consequence? Yeah.
It's a good question. So I think it's both. I think first, cancer cells have these genetic drivers and these genetic mutations that a lot of them are driving this metabolism, but also because of cancer and because of their continuous growth and the environment in which they are, they also increase their metabolism and alter their metabolism. So it's a combination. It's because of, well, yes, I think it can be both, yes. It's because of the cancer cells and their requirements that they increase it. So I would say more a consequence, maybe, yes.
Thank you. Yeah, I think we have to move on, but thank you very much.
Okay, thank you, Dr. Nabbe. Then we come to the last opponent, Professor Zuhause, Professor of Pathology at Maastricht University Medical Center.
Dear candidate, congratulations for this book and the work, also to your promoter team. Sertraline is widely used in anxiety disorders and depression patients, suffering of depressions. Is it the consequence that we see less cancer and less aggressive cancers in those patients treated for anxiety disorders and depressions?
So could you...
Sertraline is used for anxiety disorders
yes.
and depression patients in order to elevate serotonin levels.
Yes.
Do we see in these patients, in terms of epidemiology, less cancers or less aggressive cancers?
Highly esteemed opponent, thank you for your question. So we are now doing a retrospective study to see first the toxicity, but also the outcome in lung cancer mostly, but we will get the results in 2 weeks, so I don't have the results yet. So we don't know yet. We have been trying to indeed check retrospectively to see the outcome of these cancer patients, but in a lot of cases, the number of patients that were taking sertraline to say something about their outcome was too little, so we could not see if it was improving. That might be a good sign.
Sorry to interrupt you, but basically the question was more for epidemiology, whether this is known, but we can discuss this later for sure. Sertraline is inhibiting SHMT1 and 2. You discussed this in chapter 3, but you did not have a look at this. Why didn't you focus on these two, or why didn't you see for the transcriptional level, or what is happening to these two isoenzymes, or what are these?
So yes, in chapter 3, we did check the expression of SHMT, gene expression, and also protein expression. So I think we did check it, but we didn't check like the activity because this was already previously published that it was inhibiting to SHMT in those cells, but we saw the increases in SHMT.
As well, 1 as 2, both of them?
Oh, yes, sorry, yes. No, we focused indeed on 2 because SHMT1 has been less, have been described to be less involved in cancer. Mostly is SHMT2 in the mitochondria to be more upregulated in cancer and not so much SHMT1. Mm-hmm. And we were also interested in blocking that one in combination with radiotherapy for the mitochondrial function.
In order to save time, I directly ask you a question to proposition number 4. You talk about the FDA approval of this drug. The sertraline is FDA approved, but not for cancer. Oh? It's just for depression and anxiety disorders. Yes. Okay, so sertraline enhances serotonin by inhibiting the reuptake. And would it anyway be sensible to test your tumors, which you have in mice, and which show much less effect in your figure 6, which you also showed here, 6b, or what is 6d? And because the effects are less impressive, just sertraline alone compared to radiotherapy. Would it be sensible to have a look at serotonin contents or expression in these tumors? Did you do this?
We didn't look at it, no. So that might be indeed something interesting to investigate. Because
why would it be interesting? Yeah. Oh, sorry.
Because indeed those cases might have like a bit of like an excess levels of serotonin. But what we do to try to prevent to have an excess of serotonin is to give the treatment every 3 days, so we give time for the serotonin to be reuptaken and not have this excess, because excess of serotonin might have not beneficial effect, like some side effects as well. But by trying to not do it daily, like patients with depression take, but more like a spread, we might also overcome this side effect of excess of serotonin. But this might be interesting to investigate, that we really managed to do that, of course.
Yeah, because it could be that they have carcinogenic effects and just oppose the effects which you see by sertraline.
Yes.
Thank you.
Sorry to interrupt the discussion. Anaís Sánchez Castillo, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and night. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and night.
Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
U kunt op plaatsen.
Anaís Sánchez Castillo. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Voys is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I confer upon you, Anaís Sánchez Castillo, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, affixed with the official seal of the university. It's my pleasure now to hand over to Dr. Kim Kampen, who was Anaïs's daily supervisor, who will speak out the laudatio.
Dear Anaïs, we first met online during your interview for the TerraNet position in Mark's lab. You were already quite experienced. You worked for a while as a research assistant in your former lab in Barcelona. Correct me if I don't pronounce it well, eh? We were quite impressed by your CV. What we didn't know is that Anaïs had a double agenda. She wanted to do a PhD in Maastricht for also other reasons, for her love, Renzo. In October 2019, we both started our journey at the Department of Radiotherapy. I was generously appointed as your daily supervisor by Mark. During the years, I got to know you better and better. I think we always could speak up our minds both ways, which was really constructive. I could get you motivated and enthusiastic on the cancer metabolism, leading to this beautiful thesis. To most people in the lab, I think you were providing them with advice, one way or another. People admire your warm heart and your endless willingness to help out when others are stuck. You have a lot of patience, at least in the lab. I don't know in private, but... and the girls from the lab even came to visit you in Malaga, in your hometown. On the outside, you're always shiny. You always shine positively, but I know you also had your ups and downs. You started in the Corona period, which was shortly happening after you started, which wasn't always easy on you. You had difficult times, and sometimes this was an emotional roller coaster for you, because you were kept not only from the lab, but also far away from your family. You always kept your motivation, and you never sat down, which shows huge dedication and accountability. So after 3 years of TerraNet contract, your contract was ending and you started another year, but you also still had to do your secondment, which was not feasible earlier due to the Corona period. So you performed in 2022 your secondment in Pierre's lab. And as always, Anaïs never really plans a lot of things, so everything has to start at last minute, but somehow she always manages and things worked out well. TerraNet, I think, was a great experience. We went to a lot of nice places. We went to amazing venues. Like in Oxford, we had dinner in the National History Museum. And I think most special was the dinner in Paris in the Eiffel Tower. Unfortunately, the day after, you were quite sick, but you still managed to present during the conference in Paris. So this TheraNet consortium was unique, provided you with friends for life, very inspiring and fruitful, and I hope we can come up with a follow-up consortium. So along your PhD, I tried to challenge you various times, intentionally, sometimes on the spot, and not to convince me of anything, but more to prove to yourself what you're capable of, to grow your confidence on your own capacities and capabilities. You had the opportunity to present your work at the biggest radiobiology meeting in Europe, which was the biggest compliment you can get for the work you've done. So even the colleagues in Leuven, they sent us photos of the figures from your review that are used by experts in the world in lectures and conferences. So it shows what you're capable of. Not only a scientist can appreciate your work, we could all see the great stuff you did by the figures you made, and also for the bigger audience to make it more comprehensible and easy to follow. So your booklet is thick and it contains quite some papers, of which published in revision or ready for submission. But somehow you would think that if you're so far at this stage, it would be easy to write your thesis. But somehow I think it was the most difficult time for you. It's basically your perfection which keeps you fighting against yourself. I hope you also learned that you can rely on others when you're stuck, to move the big elephant away from the road blocking your progress. So no one in the world can make achievements on their own. We all need each other. I believe your PhD trajectory was memorable, very special, and you learned a lot along the way. I wish we could keep you forever, but I think it's best to gain more experience and knowledge in other fields and other labs. At least we can enjoy you a few more months. It's been a privilege to work with you, and I'm convinced your family and Renzo are also really proud of you. You're a warm heart to the lab, and as a lab, we are also really proud of you. Congratulations.
Dear Dr. Sánchez Castillo, also on behalf of Maastricht University, I would like to congratulate you with the degree you have been awarded. I also congratulate your family and your supervising team. I'd like to thank the committee members for their presence here and the time invested in preparing for this ceremony. And especially, I'd like to thank our guests from outside, Professor Sonvaux, not so far away, Brussels, but Dr. Korycinski, thank you for being present, be it online from Canada. Thank you very much. Well, I can tell you that the judgment of the committee about the way you defended your thesis was really positive. Well, that you have been awarded the certificate, so that should be okay and not be a surprise. But they were especially very happy with the way you were able to cover a broad range of topics that were questions about. So I hope that you also are happy about the way the defense went. And yeah, now it's time to relax and enjoy the rest of the day and the rest of your career. Congratulations.
Thank you.
And with this, I close this academic ceremony. We are going to make some pictures here. You can stay here or leave and go to the reception area if you know where it is.
Oh, it's empty. You only give one. For a cup.